This conference will now be recorded.
Hello students. Now we are learning about anatomy of flowering plants, sixth chapter. And in this chapter, you have to learn about the anatomy and function of different tissue systems that in monocot plants and in dicot plants. So all this you are learning in this chapter. So we have already learned almost all the concepts of this chapter like meristematic tissues. The meristematic tissues means the dividing tissues. So the classification also we have learned the classification based on origin. Based on origin, it is classified into primary meristem, secondary meristem, right? First, we have learned about meristematic tissues. So in this meristematic tissues, classification has done based on several aspects. Based on origin, it is classified into primary and secondary meristems. Right. And next classification is based on function. Right. So based on function, you know that it is classified into protoderm, procambium, and ground meristem. Protoderm, procambium, ground meristem. Right. This classification is based on what? This is based on function. Next classification is based on position. Right. Based on position, it's classified into apical meristem, lateral meristem, and intercalary meristem. Right. Apical meristem, lateral meristem, and intercalary meristem. So these are the classifications of meristematic tissue. That means the dividing tissues. Next is the permanent tissues. Permanent tissues. Complex tissues. Right. Simple tissues means parenchyma, collenchyma, and sclerenchyma tissues. Parenchyma tissues, collenchyma tissues, and sclerenchyma tissues. Right. And what about the complex tissues? Complex tissues, you can see two types, xylem and phloem. Okay. Then the tissue system in plant that you can see like they are performing some general functions, and these tissues you can classify into epidermal tissue system, ground tissue system, and vascular tissue system. Right. Epidermal tissue system means the outermost covering, right? And ground tissue system means that is a fundamental tissue system, that is the main plant body. Right. And what about the ground tissue system? Ground tissue system that forms the steel region. Okay. So here you can see the vascular bundles and all other things in the center portion, right? Or the Other than the epidermal tissue system and ground tissue system, you can see this region, vascular tissue system. And here we have learned, here you can see the vascular bundles, types of vascular bundles, open one, closed one, like that and all you can see. And here the monocot stem. Next we move to the anatomy of dicot and monocot plants. So we have learned the monocot stem and dicot stem, and dicot root and monocot root, then dicot leaf and monocot leaf. So all these structures and the differences, the characters, like in the features, characteristic features of dicot and monocot stem, we have learned about the epidermal region, the difference in the dicot stem and monocot stem, epidermis. So while writing about the stem, you have to mention the epidermis, hypodermis, cortex, endodermis, pericycle, medullary rays, pith, vascular bundle. So you have to come in that order only. Okay, from the outer portion, you have to write towards inside. Okay, so you won't miss any portion. So the diagrams are very important. Keep those diagrams in mind, so you can answer all questions. The same way you have to practice this root also. Here in the root, in dicot and monocot root also, you have to write about the pericycle, then the vascular bundle, cambium, pith, then the secondary growth of that, then the cortex and endodermis. Then in leaves, what are all characters? The epidermis and the epidermal cells you need to mention. Then leaf means what you will get? You will get stomata. Number of stomata you can mention. And the guard cells, the types of guard cells. Then mesophyll cells. Then the veins. Then the bundle sheath. So all these things you have to mention in the characteristic features of leaf. Okay, so all these things we have learned, and next we need to mention about the secondary growth, the last topic of this chapter. Secondary growth. So let's see how to mention what are all points we need to learn from this secondary growth. Actually, secondary growth that is found in dicot plants. Okay, found in dicot plants. Secondary growth is found in dicot plants, and this is due to the presence of cambium in the vascular tissue. Due to cambium which is present in the vascular tissue. Okay, and here this secondary growth that is resulting in the girth increase of stem, and this is also responsible for wood formation in dicot plants. Increases girth. So here wood formation, that helps in the wood formation in dicot plants. Okay, so here the things which we need to learn: secondary growth that is found in dicot plants, and with the help of this cambium, due to the presence of cambium in the vascular tissue, this is happening, or the secondary growth is taking place, and that increases the girth of the stem, and that is responsible for wood formation in dicot plants. Okay, so secondary growth in dicot root. Secondary growth in dicot root. So let's see first this one, secondary growth in dicot root. Actually, the secondary growth in dicot root that takes place in stellar region. That means in the steel region and in the extrastellar region. Okay, this is taking place in the steel region and extrastellar region. Okay, steel region, both in the stellar region and in the extrastellar region, it is taking place. In steel region, how it is taking place? By vascular cambium. Right, here we said due to the presence of cambium in the vascular region, that's why the secondary growth is taking place. In the stellar region, by vascular cambium, it is taking place. Which one? Secondary growth. In the case of root, we are saying now Then in the extrastellar region by core cambium. Okay, by core cambium it is taking place. Clear? Next is about the secondary growth in dicot stem. In dicot stem, the secondary growth, how it is taking place in dicot stem. The secondary growth in dicot stem, here secondary xylem produced by the cambial ring, that is called as wood. Secondary xylem produced by cambial ring, that is called as wood. Okay, this is the important point in the secondary growth of dicot stem. And here the wood which is formed in a single year, that is called as annual ring or the growth ring. Okay, what is annual ring? The ring or the wood which is formed in a single year. The wood which is formed in a single year, that is called as annual ring. So by counting this annual ring, we can calculate the age of that plant. Okay. Then about spring wood and autumn wood and all. On the basis of time of formation, you can call spring wood, autumn wood. The spring wood means that is early wood, and that forms the major part of the annual ring. And that spring wood that forms plenty of xylem vessels with wider cavities, and that will be lighter in color. Which one? Spring wood will be lighter in color. At the same time, the autumn wood will be darker in color, and the cavities of xylem vessels will be narrower, and that forms a narrow strip in the annual ring. Okay, so that autumn wood is also called as late wood. Clear? And on the basis of durability, we can say sapwood and heartwood. So sapwood, that is not durable, and heartwood is durable. Okay, sapwood is not durable. So these are the main points which you need to learn in this chapter. We have covered almost all the important points. Next, we will move to previous year questions. Here, we have learned about tissue and tissue system. You will get questions from 3 topics of this chapter: tissue and tissue system, then about the internal structure of dicot plants and secondary growth. Then third topic is the internal structure of monocot plants. So all these topics, dicot and monocot plants, internal structure, then the tissue and tissue system, and the secondary growth. These are the topics from where you can expect the questions. So let's see some questions from this portion. So first question, like all the general questions I'm mentioning here, and I'll explain the reason also. Stomata in grass leaf. Stomata in grass. So about this, we need to learn. Stomata in grass leaf, this is a neat question. See, stomata in grass leaf, that is dumbbell shaped. First point is dumbbell shaped. Okay. And in grass, see the epidermis of all green aerial plants, they have minute openings, right? These openings are called as stomata. And these stomata, this minute opening which is called as stomata, that is surrounded by which cells? Guard cells, right? And this kidney shaped or bean shaped guard cells are present in dicots. But in monocots, this guard cells, generally we will say it is kidney shaped or bean shaped, right? But kidney shaped or bean shaped guard cells you can see in dicots. But here in the monocots like grass and all, they are dumbbell shaped. In monocot, example we can say grass, in that and all it is dumbbell shaped. So here stomata in grasses. So stomata, you should know what is stomata. Stomata, it is surrounded by guard cells, and these guard cells will be dumbbell shaped in monocot. So grass is a monocot. Next question is, vascular bundles in monocots. Vascular bundles in monocots. See this question also, this is a 2015 question. So here what I'm doing is the repeated questions in several years. These questions are repeated, and all the points about these questions I'm explaining here so that you can connect with all the questions. Okay, while answering the previous year question paper, you can connect with all the questions. So almost all the important points about this topic I'm covering. So here the vascular bundles in monocots, they are closed, right? So the vascular bundles of monocots, they don't have a layer of meristematic tissue. They do not have a layer of meristematic tissues. Meristematic tissues. That means they meristematic tissues, which is the layer of meristematic tissues or continuously dividing tissues, that is cambium. So they don't have a layer of cambium, and where this cambium will be near the vascular bundles, right? Therefore, they are called as, if they don't have a cambium or they don't have a layer of meristematic tissue, they are called as closed type. That's why I said they are vascular bundles in monocots. They are considered as closed ones, right? And here, due to this condition, secondary growth is not seen in monocots. Okay, secondary growth is absent in monocots. So here, actually, we have three questions here. Vascular bundles in monocots, they are closed. Okay, and another question, what is the reason if vascular bundles are closed? Means they do not have a layer of meristematic tissues or continuously dividing tissues or cambium. Okay, and this questions two times this came. Okay, and due to the absence of this cambium or this meristematic tissue, secondary growth. Here again another question came, like vascular bundles in monocots, they are closed. Due to that, they have given some reason. In that, the best reason, the secondary growth is absent. Okay, that is the correct answer. Secondary growth is absent. Okay. Then another question is the arrangement of vascular tissue in a heterocentric vascular bundle. Arrangement of vascular tissue in heterocentric vascular bundle. Okay. So let's see here. In a heterocentric vascular bundle, the arrangement of vascular tissue is concentric. Okay, in heterocentric vascular bundle, arrangement of vascular bundle or vascular tissue is concentric. Okay, so in this, the two types of vascular tissues, that means phloem and xylem, that surround each other. Okay, so here the heterocentric vascular bundle means they are concentric type, and here for phloem and xylem surrounds each other. Here the xylem forms the solid core. Okay, and the phloem surrounds it completely on all the sides. Okay, xylem forms the which part? Xylem forms the solid core, and phloem surrounds completely the xylem on all the sides. This surrounds, right? And here you can see a strip of vascular cambium. Strip of vascular cambium that is present. Okay, so all these are present in the heterocentric bundle. Okay, and you can see this in some aquatic angiosperms and in the staminal bundle of so many dicots. Okay, so here all these 3 points, they are previous year questions. Okay, next question comes. See, which one of the tissue provides maximum mechanical support to plant organs? Sclerenchyma, parenchyma, collenchyma, arenchyma. Among this, which is providing maximum mechanical support to plant organs? See, sclerenchyma, we know that sclerenchyma. We know about sclerenchyma. Sclerenchyma is providing maximum mechanical support. Mechanical support to plants, or mechanical support or mechanical strength to plants, right? And we know that sclerenchyma, which is another point you need to learn, sclerenchyma, that is a dead mechanical tissue, dead mechanical tissue, right? And these dead mechanical tissues and the cells of these dead Mechanical tissues are long, or they may be short or narrow, but their walls are thick. Okay, they have thick walls and they have lignified walls. Lignified walls. So that is giving the mechanical support. So it is acting as a versatile mechanical tissue. The cells are dead cells, but they have a very thick wall and lignified walls, which is providing the mechanical strength. What about collenchyma? Collenchyma. Here, the collenchyma that is considered as the living mechanical tissue. Living mechanical tissue. Okay, that cells contain chloroplast. Okay, the cells contain chloroplast. Okay, and that is taking, see, chloroplast is there means definitely we can say that is taking place in photosynthesis. Okay, it's taking place in photosynthesis. And being that living cell, it is or the cells are also storing food. Storing food. The cells of this collenchyma tissue, they are storing food. Okay. Then we can say in hydrophytes, they have air and chyma, right? The characteristic features or the hydrophytes are characterized by the presence of air and chyma, and these plants which is growing, or the aquatic plants which are growing in the aquatic areas, they are called as hydrophytes, right? And the air and chyma is the parenchymatous tissue, and that is containing air space, and that increases the buoyancy and that allows the exchange of gases in the aquatic plants. So in hydrophytes, you can see aerenchyma, which is the type of parenchyma which is having the air space, and that air space increases the buoyancy, and that allows the exchange of gases in aquatic plants. Okay. Then about lateral meristem, we have another question in 2013. Lateral meristem. This question is in 2013. So lateral meristem, examples of lateral meristem. First, that's the first question. Phalogen and fascicular cambium. Fascicular cambium and phalogen. These are the examples of lateral meristem. This phalogen is Phylogen is otherwise called as cork cambium. Okay. So these questions, this kind of questions you can expect from uh, the tissues and the tissue system. Next, when we are moving to the internal structure of dicot plants and the secondary growth, the second topic, the internal structure of dicot plants and secondary growth. So from that, first we'll start from the dicot root question. This is the first question in this topic, 2019. This question came. So in dicot root, the vascular cambium that originates from the tissues which are located below the phloem bundles. Tissues below the phloem bundles. Okay, and above the tissues which is above the protoxylem. So here you can say that is originating from tissues which are located below the phloem bundle and a portion of pericycle tissues above the protoxylem. Pericycle tissues above the protoxylem. Okay, you should remember the structure of dicot root. So here the vascular cambium is originating from the tissues which is located below the phloem bundle and the portion of pericycle tissue above the protoxylem. And here vascular cambium, that is the meristematic layer. We know that the cambium, that is the meristematic layer. So vascular cambium is the meristematic layer and this layer is responsible for the cutting of vascular tissues, that means the xylem and phloem. And in young stem, this is present in patches. Okay, in young stem, it is in patches. Okay, the vascular cambium. Okay, which one? And the secondary xylem and phloem in dicot stem. How it is formed? This is another question. In 2018, the secondary vascular tissues, that means the secondary xylem and phloem. Secondary vascular tissues. About this, in 2018, you got one question. This is secondary xylem and phloem. Secondary xylem and phloem in stem. These are formed by vascular cambium. Formed by vascular cambium. Okay, and this is produced by two types of meristem. This is produced by two types of meristems, that is fascicular or intrafascicular and interfascicular cambium, produced by interfascicular and Intravesicular cambium. Okay, here the intravesicular and intervesicular cambium. This intravesicular cambium, we'll call that as vesicular cambium. Okay, so here the points. First, we need to learn the secondary xylem and phloem in stem that is formed by the vascular cambium. And this is produced by two types of meristems, intervesicular cambium and intravesicular cambium. This intravesicular cambium is the primary meristem. Okay, intravesicular cambium, this is the primary meristem, and this is in the form of strips in vascular bundles, and that will divide to form the secondary xylem towards the inner side. Okay, and intervesicular cambium that arises the or that arises secondarily from the cells of medullary rays. Which one? This intervesicular cambium. This is from the medullary rays. Okay, with the help of diagram, if we are learning, means it will be very easy for you. There won't be any confusion. And we said about, again in 2018, you got another question about the Casparian strips. Casparian strips, these Casparian strips also, they are found in the endodermis of root. In endodermis of root, you can see the Casparian strips. And this is a band of thickening strip. Okay, and this will run along the radial tangential walls of the endodermal cells. Okay, and we said one important point, this Casparian strip, this is made up of suberin and lignin. So this is one question where you can see the endodermis of root, and you can answer one more question, and it is made up of suberin and lignin. Then the third question from the Casparian strips, the Casparian strips prevent plasmolysis of endodermal cells. Prevents plasmolysis of endodermal cells. See, if they are giving 2-3 statements, you will get confused. Okay, Casparian strips prevents plasmolysis of endodermal cells. This is one important. So all these three questions came in the previous years. Okay. Then another question. About tylosis. See, the tylosis, they are balloon-shaped structures. Balloon-shaped structures. This is 2016. Okay, 2016, one question came, and you got so many other questions. The balloon-shaped structures, that is one point you need to learn about the tylosis. So tylosis, they are the balloon-shaped structures, and these structures are found in the woody tissues of dicot stem. In woody tissues of dicot stem. These are found in the woody tissues of dicot stem, and here these are the extensions of xylem parenchyma cells. extensions of xylem parenchyma cells into the vessel elements. Okay, so xylem parenchyma cells, this will extend into the xylem vessel elements. Okay, so these are the extensions of xylem parenchyma cells, and to where it will extend to the xylem vessels. Okay, and that extension blocks the lumen of the xylem. Extension procedure that blocks lumen of xylem. Okay, so these are the 4 questions of previous years from the point of tylosis. From this topic, tylosis, they are balloon-shaped structures, and they are the woody structures or woody tissues in dicot stem. And why it's become so woody? Due to the extension of xylem parenchyma cells into the xylem vessel elements. Okay, due to this extension, xylem lumen of xylem, that is getting blocked. Clear? And here, one question in 2011. The difference of old dicot root from dicot stem. Old dicot root from dicot stem. Okay, so here let's see. See, old dicot root, how it differs from the dicot stem. Differences we need to mention. Okay, root differ from dicot stem in the absence of primary phloem. Primary phloem. In which one? In old dicot root, primary phloem won't be there. So it is absent in old dicot root. And here the cambium forms the secondary phloem towards the outer side and secondary xylem towards the inner side. Cambium. The secondary growth is taking place, right? In which one? So cambium forms the secondary phloem. And secondary xylem. Right? Secondary towards outer side and secondary xylem towards inner side. Right? Old roots, primary phloem is usually crushed in roots. Primary phloem is crushed, xylem and the secondary xylem all are present. In which one? In stem. In old root, primary phloem is crushed. Okay, old root. In dicot stem, primary phloem is there, secondary phloem is there, cambium will be present, primary xylem will be there, secondary xylem will be there. Right? So all these are present in which one? In the stem, in the dicot stem. Okay. Next questions are from the internal structure of monocot plants. See the regeneration of damaged growing grass. How it is happening? See this, due to grazing and all, grass is getting damaged. So how the regeneration is possible? The regeneration of damaged growing grass. That is due to intercalary meristem. Okay, intercalary meristem. Okay, and uh, it is a meristem which is between the mature tissues. That is present. This intercalary meristem, this is between mature tissues, right? And this regenerates a part which is damaged by the grazing herbivores. Clear? So intercalary meristem is working, and that is helping for the regeneration of the damaged growing grass. Okay. Then the characteristic features of monocot root. This is the first question, and the second question. All these are repeated questions. Okay. Slight changes will be there in the questions. Still, the concept will be the same. So the characteristic feature of monocot root. Monocot root. That is the presence of vascular system without cambium, vascular bundle without cambium, right? So the vascular bundles, they are alternate and they are arranged in the form of a ring around the central pith region, and around that vascular bundles are arranged in which form? Radial form. And the vascular bundles are alternate vascular bundles you can see, and they are radially arranged around the central pith region, and here the cambium is absent, right? Here the xylem and the phloem bundles, they are separated by a narrow strip which is called as conjunctive tissue, right? Conjunctive tissue. See, now I'm saying nearly 5 questions. Vascular bundles in monocot root, they don't have the cambium, and central pith region is there. Around that, vascular bundles are arranged, and this vascular bundle arrangement in monocot root, that is radial form of arrangement, and vascular bundles are in alternate form. And here conjunctive tissue. That means phloem and xylem are separated by narrow strip which is called as conjunctive tissue, and this does not take part in the cambium formation. So the vascular bundles in monocot roots are closed and they do not show any secondary growth. Okay. So how many points here I'm saying? Monocot root, vascular bundles are present and cambium is absent. There is no cambium in the vascular bundle. So the vascular bundles are alternate and they are radially arranged in the form of a ring around the central pith region. Okay, and xylem and phloem bundles, they are separated by a narrow strip, and that is called as conjunctive tissue, and this conjunctive tissue is not taking part in the cambium formation. So there is no cambium formation, so no secondary growth, because vascular bundles in monocots are closed and they don't have a secondary growth. Clear? Here, another question is, desert grasses often roll their leaves due to the presence of which cells? Desert grasses often roll their leaves due to the presence of. In desert grass, you can see which cells? Bulliform cells. Bulliform cells. Okay. So they roll their leaves due to the presence of bulliform cells. Rolling of leaf due to bulliform cells. See, bulliform cells are big sized cells. Big sized cells, and they are thin walled cells, and they have large vacuole. Okay, they are large vacuolated cells. Okay, and they will be present towards the lower epidermis. These bulliform cells are helping out their leaf. They are bicules, and these bulliform cells are present at the lower epidermis. And this questions, in 2009 you got one question, and in 2011 all these three questions came from this part 2011, and in 2015 you got these three questions. Okay, in 2007 also, bulliform cells, they are present in the upper epidermis of monocot, like wheat, maize, in all these cells. Okay, so these are also called as motor cells, and these cells are helping in the rolling of leaves in dry climate. For what? It is for the preventing of water loss. So bulliform cells in the desert, it is present in the lower epidermis, and they are helping for the rolling of leaf. So mainly it is helping for the rolling of leaf, and they have and another question, in monocot roots, which type of vascular bundles are found? We have already told the monocot root structure. So here, monocot root, you know that monocot roots have the vascular bundles which is radial, and they have exact xylem. Okay, exact xylem means protoxylem elements lie at the peripheral side. Protoxylem will be towards the periphery and metaxylem in the center. So that condition is called as exact xylem. Clear? So these are the main questions. So while learning one question, try to cover almost all the points about that question. Okay, about the topic. See, if you are learning about bulliform cells or you are learning about monocot root, try to learn complete points. Clear? So that's all about this chapter. Thank you.